Okay, DOM. Venn diagram. Inorganic carbon. Inorganic meaning like carbonate system, like carbonate.
Sure, yeah.
Bicarbonate.
Yeah, I see it as well.
Yeah, I'm gonna write it all out.
There's probably overlap with dissolved gases and with suspended particulate material and all kinds of things.
Large, I mean.
Crazy metal.
Great metal drum. Detritus, I'm just gonna write detritus of all. Because that's gonna
Catch all, yeah.
Awesome. Okay, so clearly there is a lot of stuff in the ocean that you might want to measure, and some of it maybe not as easy to measure directly with optical methods. And the manual is going to connect with her to talk about what we can, what are the things that typically directly fall out of sunlight stuff. Some stuff we have to infer processes with coupling optical measurements to other measurements that maybe are going to directly get at the same time. So it's just important, I think, to start off with this mental picture of what might be paramount, and then how we get there from these kinds of tools. Okay. So the next few slides are going to just sort of go through some things to think about as we start to think about how light interacting with all these particles and constituents of the alien. There's a whole bunch of stuff on the slide, so I'm going to ask you to just look inside to start with. I posed a question up at the top. What fraction of particles are organic? Right? What is just something that was maybe once made by a living thing, or then aggregated or disaggregated or dissolved or whatever? But what fraction of particles are organic? As an example, I'm going to walk you through some data that were collected on an Arctic geotraces. Chemist, there's a huge program called Geotraces. It's now been collecting data on trace metals as well as other things all over the world for over a decade. This is the 2015 Arctic geotraces. Out in ice free period from August to October, they left from the little pies and mini inset map where basically they left out of Alaska and went out to almost the boundary between the Canadian and Eurasian basins, and then came back. Their cruise tracks going out and then coming back. All of the 6 plots here are all sections. So the horizontal axis is going from their starting point out to the middle and then back for 6,000 kilometers. And each of these, what they did was, among other things, they put submersible pumps into the ocean that suck up like a cubic meter of water on filters, brought this filter back, and sampled them for trace elements, which they interpret to give the fractions of that mass, right, that was on the filter that was organic. Inorganic, lithogenic, made up of iron oxyhydroxide, some fine rust particles basically, oxidized manganese, calcium carbonate, and sulfur. So these are some of our major mass constituents, just generally in the ocean. So particulate organic matter is this one, lithogenic material is this one, iron oxide hydroxide here. So it's what diatomic cells are constructed out of. Calcium carbonate, same for other things that I made. And each of these, the color scale is the same, no, it's not. The color scale, however, is showing the mass fraction of each of these states. Interesting things to call out. Near the surface, in the upper 500 meters or so, most of that material is organic, which kind of makes sense that that's where it's being produced, right? It sinks down, but it's consumed. The second panel down, this guy, the scale goes from 0 to 1 color scale. And the fraction of particles in the water that are lithogenic, right, have high alumina, we can tell they're actually ground up lithium rocks, 50 up to 100, 50 to 90% lithogenic, right? So hardly any organic matter at all. There are a few places where we've got high concentration, or like a couple of spots along continental shelves where there's high concentrations of excited particles. I'll come back to that later on in a different slide. In this particular part of the ocean, some opal, like maybe 30% opal in a few places near the surface, not a lot of calcium carbonate. And then this funky plume of about 5% of the mass is manganese oxide, predominated as the oxidized the basis food. But anyway, so I've given you this this example just to show you of all the stuff that's in the ocean, how much is organic. Okay. How much of that particulate organic matter is then alive, right? Say, well, that's a cell. That's living. It's doing something. So here is an older paper, but it's a compilation of data of microscope counts of and then estimations of the carbon contained bacteria. So that's this line here. Phytoplankton. And then this is their sum in the top line there. Bacterial organic carbon, phytoplankton carbon, and their sum. And this is the function of POC concentration, and this is a bunch of different things. Environments. And so what you see, a couple of things pop out. One is bacterial organic carbon is comparable to phytoplankton carbon, right? The cells just from the bacteria, and then oligotrophic water, where the POC concentration is lower. The bacterial line, which I'm outlining here, higher. Okay. So this is just one place, right? One area in one study, but illustrating the distribution of biomass among these two types of cells. Here's another study. This is a big modeling. Pulled together a whole bunch of different data sets to try and estimate biomass among different major categories of organisms over the whole globe. So lots of assumptions made in there, but the different colors here represent different like mammals, fish, zooplankton, autocross, phytoplankton, and bacteria. And they plotted the global biomass in gigatons versus the body mass in the different categories. So colors show the categories. So this is the volume size distribution. Same data, log, and linear scales. And the take homes are basically that Sheldon 1970 not too far off, basically flat volume of biomass distribution with log space. He did the whales, the marine mammals, are a little bit more. Other than the bacteria are a little higher, but other than that, pretty good prediction. And actually, the rest of the paper is kind of interesting to project how much more we're taking out. And the perspective of how humans are changing it based on their activities. So that's worth reading. I'm to read interesting things. But the point of this slide overall is just that I'm just trying to point, right? Not an organic matter, it's not a study.
9:55.
Okay. So next question I would pose is what fractions of delving into the optics, what fraction of particulate stack scatters is due to phytoplankton? We sort of thought about, okay, how much of other stuff is there? So the top, both of these panels are from a paper by Jason Brown in 2015. He's not the only one who's got relationships like this now, but this is one of the earliest. Basically, the upper panel is showing the scatter plot relationship between backscatter at 470 nanometers and particulate organic carbon. These were data from heat. So the top panel is BDP versus POC, and the slope of that fit about 49,000. The bottom panel is BDP again on the x-axis, and this time all the samples have gone from a sorting flow cytometer, so that measures the carbon of the phytoplankton generating the backscatter, or that were contributing to the backscattering of the overall water cycle. And here the slope is about 12,000, and more or less. I mean, there's definitely variations in what people find in their carbon phytoplankton carbon versus backscattering, but it's just another sort of not all phytoplankton. And this is a more recent paper by Felicio et al. Where they took the intercept of the regression of backscattering against chlorophyll by month in different pixels and this is the plots we're looking at here of the intercept. So it's basically the backscattering is not linearly covariant with chlorophyll in different regions. I just picked out January, July here. But the point here again is that there is an important fraction of backscattering that is really and regionally dependent. It doesn't just go because again, it's hammering home this diversity of stuff. All right. There's definitely even the phytoplankton part of that.
Once you move, once you account for average, you see that this ratio is both for that. And there's a week. That's the target. The good things we all do. Both the office and both.
Great.
So Meg just set this slide up really nicely by showing that map of some of these differences in the phytoplankton fraction of BVP. But I wanted to talk more specifically, if you're thinking about optical proxies, often you'll see this relationship between BVP and chlorophyll. And that can really vary on the scale of your observation and the dynamics of the region where you're making your observations. So on the left side, we're looking at an open ocean example from lots of different cruises in different open ocean environments. And you can see there's kind of this spectrum of the relationship between particle backscattering and chlorophyll, but that you mostly have a linear relationship. And so if you assume, all right, we have this linear relationship between particle backscattering and chlorophyll, and then you want to take that relationship that's been established collecting all these samples in the open ocean and apply it to a different ecosystem, you might get a really different result. And so the axes are reversed here, but I'm showing a different example from the Santa Barbara Channel that Fernanda Hendricks Freitas put together using glider data. And so in the Santa Barbara Channel, there's this observing program called Plumes and Blooms, because the two major sources of optical variability are either plumes of sediment or blooms of phytoplankton. And you can see that duality really well here, where you have times where there's a very strong relationship between chlorophyll fluorescence and particle backscattering, and other times where there's no relationship between those two variables. And then the third case is where you have very low chlorophyll, but really high backscattering. So that phytoplankton fraction, or the chlorophyll covarying fraction, as Meg was talking about, can really depend on what your situation is in the ecosystem where you're observing, the scale of your observation also. So these were small glider surveys, but going in one transect across the channel. So you have variability in your distance from shore. And then it really can differ if you want to apply a global open ocean relationship that might not be appropriate for your local coastal observatory. I also wanted to highlight that there are other things in the ocean that can be dependent on other ecosystem processes that we might be interested in observing. So Kelsey and I participated in a research cruise that serendipitously took place during a coastal wildfire in the Santa Barbara Channel. And I'm showing here the fraction of detrital particles and phytoplankton from IFCB images. So we used the IFCB to trigger on scattering instead of fluorescence, because we were really interested in capturing a large variety of particles in the water. We knew that there was all this ash being deposited from the wildfire, and we wanted to see if we could quantify the fraction of that ash that was in the phytoplankton size range. So because we didn't really have an ash classifier, we needed to manually validate all this data. We knew that we had detritus and ash, but we weren't able to separate those visually all the time. So I'm showing examples of what some of those ash particles looked like from this pure ash sample that we ran through the IFCB, but we ended up grouping ash and other detritus together and then looking at that fraction compared to the phytoplankton fraction within the IFCB size range. And you can see that even over the course of this week that we were out there, there's really highly variable inputs of detritus compared to the phytoplankton component. And so you might sometimes have other complicating factors in your detritus versus phytoplankton comparison in your ecosystem. I don't think that you would ever expect to see that large of a fraction of detritus if you were in the open ocean and there was a phytoplankton bloom, but maybe if there had been a volcanic eruption on an island nearby and you had this particulate deposition, that would change the balance of phytoplankton and other particles. So we've been talking a little bit about chlorophyll breakdown this morning as part of your fluorescence lab, but I just wanted to make sure, I don't know that either Ivana or I fully in detail talked about these chlorophyll degradation products. When we're thinking about this process by which phytoplankton turn into detritus, I think it's important to know some of these indicators that we might use to talk about that phytoplankton fraction. You all are very familiar with phaophytin because that's something that you're measuring in the Turner fluorometer at this point. But so if we're looking at this breakdown of chlorophyll as a molecule into different degradation pigments, there are a couple of different processes by which that can happen. So Ivana talked about the chlorophyll to phaophytin transformation. That happens when this magnesium ion in the chlorophyll molecule is kind of kicked out and replaced by 2 hydrogen ions. So you can see that that process is modulated by either acid, as you added in the fluorescence lab, or heat, which we've also talked about in the case of quenching. But there are also other processes that can happen, usually that are modulated by enzymes. So where might you find an enzyme that would be causing the breakdown of chlorophyll? Zooplankton guts, awesome, right? So this chlorophyllase enzyme is being used to break down the chlorophyll molecule into usable energy, but it removes this phytyl group from either chlorophyll, or if it's already been converted into phaophytin, from phaophytin, and then that can result in these other 2 degradation pigments, chlorophyllide and phaeophorbide. So if you look at a standard HPLC that's the results that are coming out of an HPLC instrument meant to measure phytoplankton pigments in the ocean, you usually see chlorophyllide, phaeophorbide, and phaophytin. And so if you're interested in your particular system in characterizing the fraction of those different pigments to chlorophyll A, characterizing their concentration over the course of your study, for whatever reason, you'll be able to get the concentrations of those pigments and look at them in ratios. So also thinking about the breakdown of phytoplankton and its transformation into detritus, I'm just showing this example of a microscopy image of a sediment trap gel. And so you can see all these different particles that are falling into this gel are all things that in the ocean we might either consider living or dead. So Meg kind of set up this motivation of we have living particles and dead particles, but what's their balance to each other? I can tell you that in this type of sediment trap collection, you have sinking phytoplankton cells, you have zooplankton fecal pellets that contain traces of phytoplankton, you have zooplankton cells themselves. All of those might be grouped into a living fraction based on their contribution to maybe say a chlorophyll fluorescence signal, or a dead fraction based on their contribution to particle backscattering or POC. So when we're thinking about all the different ways that phytoplankton are transforming as they sink out of the surface, I'll refer back to that geotraces figure that Meg showed, where we had the really high organic fraction in the surface. And then as it's sinking, it's becoming more this inorganic fraction and combining with the lithogenic fraction to be kind of removed from thinking about that organic carbon, living phytoplankton, and more into a detritus.
But from your perspective, or, based on what you might guess, what constitutes what should be defined as boundary particle? Say, here's the particle, here's not the particle. Index of refraction, okay, or change in the index of refraction. That's very sort of physical optics definition. And that's so meaning like very close within triangle. Yeah. Density. Had your hand up a little while ago. All right. Yeah, I mean, they're all valid. Right? Just to sort of get you thinking, here's some examples. Particularly of more of these particles, boundaries are less well defined. So the image I'm showing up here is a transparent exopolymer particle aggregate. So this is a mixture of subparticles that are bound together sort of amorphously by polysaccharides that are exuded by diet. So they're sticky, they use this probability that particles, once they run into each other, will stay adhered to make aggregates. There are blue bits in there because it's stained with also blue stain that sticks specifically to that acidic polysaccharide matrix that holds this aggregate together. So this is kind of a watery particle, often difficult to see the edges of the gel. It doesn't backscatter a lot. Here's another one. This is a scanning electron microscope. And it's very tiny, scale bar of 1 micron of hydrothermal plume particle in specific rise coming out in the buoyant plume. And it looks like there's kind of these teeny little particles that are all stuck together like a big huge bunch of microscopic grapes, right? Those are little iron hydroxide and particles that show up sometimes. Right, this is another aggregate on a very, very good. And then finally, this is the diatomic which I think we already saw. Here we've got a bunch of particles with probably more clearly defined edges above, but in a chain, right? So there's boundary of one cell and the boundary of the next cell and the boundary of the next cell. So what is our optical? Right? So so there's sort of overlapping definitions of what is the particle. What do you think about it? Okay, so we just spent like half a lecture talking about the ambiguity of some of these, things in the ocean that we might want to try and use, optics to sense. So, what are the advantages of using these methods? Well, one is they're synoptic optics. You're using remote sensing, orbiting sensors. You can look over large scales at the, right? Look at time changes, over large stations. There's something about a way to access that. Huge advantage there. A lot of optical methods, will also be high depth. For example, we're going underwater continuously, compared to you have to get bottles. Right? Fire and forward them. Water, right? So there's a lot more information you can get on very fine scales. And with remote sensing, and some of these optical sensors, you can also either directly or synthetically, by adding up a lot of images or a lot of samples, scale up your sample volume. So if you've got something rare that you're trying to access, like a type of particle that there just aren't that many of, you can use sensors like this to scale up and start to do a better job of quantifying the kinds of rare particles. And then finally, we can, go ahead. So you can't quantify a single particle, but the remote sensing image gain information that came from all of the rare particles. Okay. So if you take a liter of water, you're changing some kind of fecal pellet or a goat kind of itself, right? That's pretty easy to look. Volume, but if you look at the whole, you know, not drawing a blank, hundreds of square kilometers, right? Yeah. You might not be able to correct using that those kinds of right? Add it up with it and all of those.
Yeah.
I know that.
We'll
That's a lot.
ask Taff Mitchell before she comes.
Yeah. Exactly. Yeah, so that's what I can. That's so clear. The last bullet point up here is just noting that we can take this, incredible properties of optical remote sensing methods to cover large distances at high resolution, good time resolution, maybe the less ambiguous and more detailed discrete sample analysis, right? That will tell us where we're looking at, right? So anyway, these are some advantages, and we'll come back around to this at the end of the exercise that you did today.
Yeah, I'll just say to that last bullet point, as when Meg and I kind of tasked ourselves with talking about this, we came up with too many examples of things either that we know of that have been done in this way, where you're relating things we can optically detect and things that we can measure at higher resolution, or the things that we wanted to be able to do that with. So maybe a way that you can think about this next part of the lecture is kind of to think creatively about things that you might want to measure, and if there's an optical proxy that you could relate to it or not, and then how might you overcome that challenge? So just kind of keep that in the back of your mind, and you'll think about it more later this afternoon and have that brewing.
Thank you, fix amount of money, even though we didn't do it a lot.
You can maximize, yeah. Yeah. Optimize those two amounts of money to one bigger result.
Manager for Okay, so the first example is first a little excerpt from a modeling paper, and then how that got turned into the longest sample program. based on remote sensing. So, so this is a figure taken from a paper that Dave Siegel published in 2014. He made a food web model driven by remote sensing observation. And the food web included things like zooplankton, freezing mortality on diet. Where do people call all these things that you can't see direct sunlight? It's going to be responding to the phytoplankton. So these two plots are showing modeled rates of grazing mortality on small phytoplankton and large phytoplankton, nanoplankton and microplankton. And he arrived at this by looking at the rate of change of net primary productivity modeled from phytoplankton related things in the satellites, and looking at changes and what wasn't there, basically. So looking at the disappearance of that primary production and phytoplankton biomass is able to infer mortality from grazing phytoplankton on these different size classes of phytoplankton. So it's sort of it's this method of inferring something not accessible. And then this was reworked into or part of the rationale for the massive content export program. The goal is to quantify mechanisms for ending the biological carbon pump and to predict that from remote sensing, right? So from things that a satellite can see, what material is being transferred into the twilight zone, right? Well below the photograph. We're never gonna have satellites that can look down there. Probably. I don't even think LiDAR is gonna be that, right? Maybe. Okay. So not like transfer to, you know, yeah. So anyway, so this is just sort of the example is that you can make remote sensing measurements of perhaps these parts of the food web. And by looking at how these different components of the food web change over time, you can get to all of us. That's the goal. Still a lot of work to do. You have amazing data to do it.
Yeah, so thinking about then this motivation that there are things that we want to be able to detect in the ocean, and remote sensing gives us these unprecedented spatial and temporal scales. How can we put together these observations that we have that are not very well distributed in the global ocean based on the necessity of the difficulty of going out on a boat and the limited amount of time and money that people have to make these observations with unique spectral signatures that we'll see in the remote sensing data? So at least when I talk to non-science family members or friends about looking at the ocean with satellites, a lot of time the first thing they ask me is, like, can we see plastic in the ocean with satellites? Because I think a lot of people know that that anthropogenic marine pollution or debris is a huge problem, and we're concerned about the distribution of plastics, whether macro or micro scale plastic, and we want to know where they are in the ocean and if there are ways to clean them up or ameliorate that situation. So I'm showing example from two totally separate papers. The one on the left is showing observations from ships of the density of this anthropogenic marine debris. So this is either kind of ships of opportunity, like taking advantage of boats that are out anyway and making observations of floating plastic, or research cruises where part of what they were doing was making observations of the plastic that they saw, but it's usually larger floating debris. So it's not microplastics. That's definitely a very important issue, but kind of a separate issue from this. And then this 2020 study was looking with Sentinel data in the near coastal region where they could go out and validate what they were seeing on the same days and the same region where these images were being collected, and dividing these different things that they were seeing floating on the surface of the water, these kind of macro scale things, into these different categories, and then modeling how they were expected to impact the optics and comparing that to how they did impact the optics. So they developed this on the y-axis floating debris index, which is where you know that you have something big in the water that's big enough that you can detect it with a satellite image that has high spatial resolution. And then they compared it to the NDVR, which is something from terrestrial remote sensing that they use to look at trees basically, and like different types of vegetation versus different surfaces in places where you're looking at vegetation next to other surfaces. So you would expect to maybe find a different spectral signature from seaweed than you would from plastic, than you would from ocean water. And in fact, they did find this really clear separation of these large scale floating objects. So I know there are a lot of people working in this space to kind of put together these spectral signatures of large plastic or microplastic, but I didn't find anything when we were looking through things the past couple of days that has combined these two approaches yet. So maybe once we have slightly higher satellite resolution, we'll have more capabilities to develop a floating debris index for larger areas of the global ocean, and then try to identify these regions where large floating debris are a really big problem. Yeah. Yeah.
So I don't know this paper, but looking at the scales for plastic.
Yes.
It's on the bottom. Oh, it's seawater. Okay, never mind. Got it.
Oh, yeah. So, right. So it's really clear to distinguish everything else from seawater, but distinguishing other things from each other is more challenging. Yeah. Awesome.
So this is another institute. Example of use of an optical sensor collecting data at really high times as a function of depth in order to quantify spikes in the signal. So this is the profile I'm showing you here from a paper with Chris where this was data from the North Atlantic 2008 experiment, and they noticed on their gliders and floats they had very high frequency recording backscattering fluorescent sensors, but the profiles were really spiky. Right? And one thing you could do if you have a very spiky profile is, filter it to get rid of the spikes. That's not signal, right? Well, Nathan and his advisor Jim Perry asked, well, maybe that's a signal, maybe that actually corresponds to something. What they found by quantifying the height and of these spikes in the profile, which I guess the black line is the filtered smooth signal, and the gray line is the spikes above that baseline. By tracking these spikes in subsequent profiles in a Lagrangian experiment, they're sort of following the parcel of water they were actually able to relate these spikes to speaking plumes of particle radiation. So this was signature of large particles, large enough that when they pass through the backscattering through sample volume, they cause a transient spike, right? Those are caused by these large particles. And this other figure came from a more recent paper from Nathan and his colleagues, where they took profiling floats now all over the world and looked into these high resolution profile series that they collected to find basically plumes of sinking spikes. So they were able to identify many of these in various places in the world. And they were able to show not only that there is a plume of sinking spikes, so the example one in the North Atlantic illustrates it pretty well in the upper right-hand corner, where there's the green, each of the green dots are spikes from these profiles. They were able to sort of fit a line to that in time and extract the sinking velocity. They also found that as you go forward in time, with the plume arriving at depth, there's this appearance of scattering or in this case, chlorophyll fluorescence in the filtered part of the signal. So that would be the black line here. And they interpreted this as a disaggregation of these signals. Okay, so in this way, they were able to use this really high resolution capability of optical sensor platforms. They did have a process that really kind of observed very well, certainly not in this sort of widespread way. Okay, so then our last thing that we want to talk about in terms of advantages of optical sensor is how we can combine these methods with high information content, but we can't do that many samples types of methods that are out there. And I wanted to just point out an example we're going to talk about here, distinct from proxy. We're going to get into later this afternoon and then in subsequent lectures later on in the course. But you could think of them as what might be a very early stage. How can we look at optical and remote sensing data and connect them to interesting. So I think I've got a deep ocean example, a CDOM, a fluorescent CDOM example. So this is actually a graphical abstract from a paper from Aaron Stubbins back in 2014. I often don't submit graphical abstracts to my papers where it's an option because this is actually a really great one because it very clearly illustrates what the paper did. So the right-hand bit of it is similar to the Venn diagram I showed you guys about DOM and CDOM and FDOM, right? So it's just illustrating that fluorescent dissolved organic matter is a very specific component of the total DLM tool. So they took fluorescent, or they took water samples and they measured the excitation emission fluorescent spectra of their samples. So they were just looking at the fluorescent part. And basically this is, we talked about single wavelength excitation, single wavelength emission for CDOM and for chlorophyll so far. But you can scan through sites sequentially at high, as a function of wavelength, and discrete wavelength bands, and measure emission as a function of wavelength in discrete wavelength bands, and you can get this three-dimensional fluorescent spectra, excitation emission matrix. And so this little green or blue box with the contour in there is basically emission excitation wavelength on the y-axis, emission on the x-axis. So those data can be decomposed to give the contributions of characteristic fluorophores that you sometimes see in dissolved organic matter. So they did that, and then they also same samples, which of course contain a lot of organic matter that doesn't fluoresce, and they put it through an extra high resolution mass spectrometer, Fourier transform ion cyclotron resonance mass spec. Measures the mass of the molecules going into the mass spec at such high resolution that you can get exact chemical formula of every molecule. You may not know the structure, you may not know where it came from or what it's for, but you can get the formula. And so you can plot the characteristics of those formulas. In this case, it's the Van Hebbelin plot that's got the oxygen carbon ratio on the x-axis, carbon ratio. And you can start to get centric distribution. Incredibly complex molecular spectrum. That's the end of the very high information content and very low sample density part of this example. So they took all their samples, they did their decomposition of their beams spectra into different fluorescent components, and they related those. So each colored line in the plot here in the lower left is one of these components up here. The example I'm showing you only shows 5, which you can get more than that sometimes. And they related those to the molecular weight in daltons or grams per mole of all of the compounds that came out from this sample. So this is, like I said, an example of maybe the very beginning of proxy building. We don't know what those molecular weights correspond to, we don't know what molecules are compared, or how they're related to fluorescence. But the fluorescence is a heck of a lot easier to measure. You know what you need, right? Well, be careful about it, but it's a much faster and cheaper than putting something into an SPSR. Right? So there's kind of a beginning there. What time is it right now?
10:30.
10:30.
So 31.
So the other example I'm going to give is another hydrochemical. So this is a figure from a paper of mine. Just a cruise of opportunity I did with Scott. This is what optical properties you could measure in black smoker fluids, right? So these are indicators of hydrothermal events, black fluid coming out of it. That's iron sulfides that then slowly reoxidize. What we did was we had a CTD with the standard C star and some point turbidity. All I had, neither one was calibrated. For the sensor. I tried to find the calibration information for the turbidity sensor. I just took voltage, and normalized voltage, and the beam attenuation coefficient, high, middle, which made an index. So this left-hand panel is basically beam attenuation measured from that CTD, C star. So the gray line is the down pass, black line is the up pass, and it looks different because one, the package moves relative to not necessarily going down and coming up to the same place. And then the other part is when you drag a CTD rosette through anything that's happening, might be disturbing, right? So might have a nice clear plume structure, and then you drag your CTD through it. So that's why they look different. We fired bottles on the up pass with a white circle, and then I took the samples and I measured them in AC9 of how the AC9 lights were still partway through, so I don't have absorption measurements, but I got good beam C measurements. And they matched really well, which was good because it told me that getting bottles wasn't necessarily changing things optically a whole lot when bringing them up 5,000 years. And the AC9, of course, in the list where I looked at bottle samples didn't go down to the fully they're not rated for 5,000 years. So I had to use them on the chip. So this is beam attenuation. A couple samples where we're overlapping in the AC9 in the list show fairly consistent, so that was also reassuring. This is a log scale, by the way, and that's the ground. Really high. I don't really want to do all that, but it was only a 5 centimeter. So we also have estimates of particle size distribution, slope, limits, again, really high numbers, really tiny particles, just precipitating out of water in the center panel. And then the thing I really want to show you is on the right hand panel, which is the ratio of the list or the LSS, the side scattering turbidity sensor, to the C star on the CTD. And I normalize them so they're equal to each other, or there's almost no scattering. And I'm just going to highlight features that are important here without going into exactly how I'm showing the uncertainty. These red peaks are all places where the turbidity sensor is spiking relative to the beam attenuation sensor. And the argument we make in the paper is that this is really for iron oxide. So this was just a hand-wavy conjecture kind of paper. I didn't have all the data I needed to really do it well. Later on another geochrome from across the South Pacific Gyre. This is not my data. This is from Dan Onemus and Phoebe Lamb and Ben Twiney. Geochemists. They measured. They also had a C star and a turbidity sensor paired together. They did a better job with their calibrations. Turbidity sensor. So they got the turbidity to beam attenuation ratio, and they ranked their samples by turbidity to beam attenuation ratio in this bottom panel here, so from low to high. And the color scale is given here. So these are the same kinds of compositional categories that I showed you that first example of Arctic, right? And so for most of this transect, in the upper panel here, we're going from about 75 degrees west up to 160 west across the South Pacific Gyre, to pass over the East Pacific Rise and along the way. For most of this, for most of these samples, the ratio doesn't seem to respond all that much to particles, right? But right here at the very high end, where they have these very high turbidity to beam attenuation ratios, there's this big chunk of ions. So it's sort of another, they did a much better job. Again, beginning of a process. Sensor package down to start tracing those sorts of particles that are probably coming out of this high temperature. Okay. Yep.
All right, so the last example we wanted to talk about is maybe not so much developing a proxy as confronting a proxy with this rare data. And I'm not going to get fully into detail on this because I'll leave this to Allie Chase when she comes to talk to you all later this month. But Allie had a really great approach with taking multiple different ways of looking at phytoplankton community composition, both from the IFCB and from pigment. So you can see that on the figure on the left, comparing the diatom carbon from the IFCB to diatom carbon from pigment-based methods. So you're making a lot of assumptions to convert that pigment to carbon. You're making some assumptions to convert IFCB images to carbon. And you can see that there is a relationship between those two properties. But then the step that links it back to remote sensing and optics is that she looked at a lot of, showing here 4 different examples from other work and then the linear fit for her data, the relationships between chlorophyll and diatom carbon. So since chlorophyll is something we know we can really reliably get from remote sensing in most places, it's a great opportunity to look at if there are relationships between chlorophyll and phytoplankton community composition. In this case, Allie took multiple different satellite-based methods that are looking at chlorophyll concentration and relating that to diatom carbon, and then confronted it with her really high-resolution IFCB diatom carbon data. And so the way that I love to look at this figure is to just choose a chlorophyll concentration along the x-axis and then follow a straight line up along the y-axis with your eyes. You can see that at the same chlorophyll concentration, there's orders of magnitude difference in the diatom carbon that you're getting at that exact same chlorophyll concentration. So as we're thinking about developing these proxies, all these samples were collected in the North Atlantic over different seasonal cycles. It took a huge amount of effort to manually create a classifier and then validate an automated classifier and get all of this information. If you have to decide a way to optimize the samples you're collecting versus the remote sensing data that you're using, Emmanuel brought up this budgetary constraint. You also probably have some time constraints and some logistical constraints. If you can develop over time data sets like this that span a huge variety of conditions, then you can start to look at whether the proxies that you're developing to be cosmopolitan for areas of the open ocean are actually working when you look at them compared to the data. Again, Allie will talk more about this when she comes and talk a lot about remote sensing methods for phytoplankton community composition. But when we're thinking about things that we want to detect in the ocean and different types of particles impacting the optics, there can be a lot of variability in what that signal looks like and the assumptions you're making to develop those proxies. I think that's almost done. Yeah.
Wrapped up our tour, particles in the ocean examples. And now we are gonna go, Manuel's gonna talk to you about particle characteristics and how they influence photosynthesis.
Thank you.
Let's just bring it together.